Thanks so much for the opportunity to present on a topic we're really excited about today. So we'll be talking a bit about rapid reviews with a focus on COVID and the challenges and opportunities. Next slide. So I will not read our whole acknowledgement of traditional land. Would like to highlight that RG and myself are white settlers on this land and that we're both committed towards working for reconciliation. So we will define rapid reviews, describe where you can find information on methods for doing rapid reviews, and then present a case example. So this was a COVID-19 diagnostic test accuracy network meta-analysis that was informed by a rapid review, and then talk about some challenges and opportunities. So I'll start off with rapid reviews. So this was the recent definition that was put forth by the Cochrane Collaboration Rapid Review Methodology Group. And so I like this definition because they talk about rapid reviews using accelerated methods. And so you'll be doing this through streamlining or perhaps omitting methods to produce evidence for knowledge users in a resource efficient manner. So what you're trying to do is you're trying to quickly provide evidence to decision makers so that they can make their decision within their decision making window or time frame. So on average, a rapid review would take about 5 to 12 weeks to complete. However, during COVID, this shrunk down and sometimes to 5 to 10 working days. And on average, we have these priced out as around $25,000 Canadian. And this would be in comparison to a full systematic review that, as you know, would take probably about a year to complete and cost around $100,000 Canadian. Next slide. So just to say that rapid reviews have been used for health decision making for a long time now. And so we've seen that it did help when we use this in our decision making, it has saved money. It also was incredibly helpful during urgent situations. So rapid reviews were used quite a bit during COVID. And just to say also that as a field, we are trying to move away from rapid reviews as a method on its own, and it's more of a framework to situate your knowledge synthesis in. So because you could do rapid reviews for many types of knowledge synthesis, so things like scoping, you could do a rapid scoping review as an example, or rapid network meta-analysis, as RG will be talking about in a few slides. Next slide. Also to let you know that there are different types of rapid evidence products. So there are inventories. So this is kind of like a more like a bibliography of or a list of studies that might be relevant to your topic. A rapid response brief. So these are usually when you are doing rapid overviews. So you're only focused on pre-existing reviews and you're summarizing them very quickly. You could do inventories or rapid response briefs within the matter of a few hours. And there are a lot of groups out there that provide this type of service. And to say that we're talking more about rapid reviews, and the distinguishing factor between this one with the star and the other types is that for a rapid review, you would actually do a full literature search with a librarian. And you're trying to get as close as possible to the systematic review method. Next slide. So just to go through the steps, so I think we're all familiar here, being with Cochrane, we would understand the systematic review steps. So I'm just going to highlight some differences with rapid reviews. So for example, you may only register your protocol. You probably don't have time to publish your protocol because you have to get these results to your knowledge user or decision maker as soon as possible. You may limit your literature search. So you might say, you know, with COVID, perhaps only if you're interested in a variant of concern, for example, maybe you would just limit your search to the last couple months or when that variant emerged globally. For level 1 and level 2 screening, you may just have one person do this. So you may do a pilot test, and then when everyone is in agreement on how to go about doing this, one person would screen everything versus having two people independently in a systematic review. For abstracting data, the same kind of idea. You may have a single reviewer, or perhaps you have a second person only review the most important data. For critical appraisal or risk of bias assessment, you may have one person do this, or sometimes you may not have capacity. So you may say, we'll start off, our first iteration of the report to our decision makers won't include risk of bias appraisal, but we'll do it in the next iteration as an example. You may limit the number of tables and text that you use. So very high level summary of study and patient characteristics. You may not have enough time to do a full meta-analysis, so you may just have a quick descriptive write-up or summary. And then you may have a more streamlined product, so a one-page summary as an example. And anytime you do rapid review for your decision maker, you need to discuss the policy, practice, and clinical implications with great caution, and let them know that you did take some shortcuts and you streamlined your methods to get evidence to them very quickly. So that may actually mean that they have to interpret your results with great caution. Just to say that we were commissioned by the WHO to write the guide on how to conduct rapid reviews. And so in our guide, we recommend tailoring your methods to the needs of your decision makers. And we discuss several different ways to be very thoughtful about this. So we don't want to be doing this haphazardly, and we only want to be doing this really for providing evidence to decision makers quickly. There's no other case when you should be doing a rapid review. So all of these different issues are discussed in great length in our practical guide. Also to say that we are working on a PRISMA extension to rapid reviews. So this is being led by Adrian Stevens, and that myself and RG will be working with her and others to write a grant proposal on this, to update the guideline and to disseminate this widely in the next couple months. We did write a paper on how rapid reviews were even more challenging during COVID-19. This was published in the Journal of Clinical Epidemiology. So definitely check this out. And we talk about a lot of the practical issues that we encountered as many different rapid review producers around the world. And we give you some tips on how to ensure that you're able to do your rapid reviews in time for decision making. And now I'll hand it off to RG.
Perfect. Thank you. So we'll continue with a real world example. We'll focus on our rapid review on the comparative diagnostic accuracy in terms of sensitivity and specificity of the rapid antigen and rapid molecular tests for the diagnosis of COVID-19 infection. And Health Canada and PHAC, Public Health Agency of Canada, commissioned us to perform a rapid review to determine the most sensitive and most specific rapid test for the diagnosis of COVID-19. And at the very first meeting that we had with them, they requested that we provide them with these results as soon as possible. So we had to quickly set up our team, where we considered to include policymakers who requested these findings, clinicians, contact experts in the field, research methodologists, and clinicians. And we also included two patient partners. One patient partner was a stay-at-home caregiver with 5 children. These children were aged between 8 and 17, and they actually carried out so frequently rapid tests per week, 3 times per week, if I remember well, so according to the government's recommendations. And the second patient partner was a freelance health journalist and former health editor of the Globe and Mail, and they both advised us on how to interpret the results and how we can disseminate these results. And at this point, I would like to mention that this was a collaborative effort through Health Canada, Public Health Agency of Canada, Ministry of Public Health in Thailand, and the Irish Department of Health. So as a first step, we broke down our research question into the PICO criteria, where we decided to include adults and children screened or suspected for COVID-19. The target condition was COVID-19 infection. We included studies evaluating one or more commercially available COVID-19 rapid tests, rapid antigen and rapid molecular tests. We used studies that have used the reference standard, the PCR as the reference standard. We included RCTs and observational studies providing us with data, and the outcome was sensitivity and specificity of the rapid tests. So we registered our protocol with Prospero, and we followed the PRISMA-P guidelines and the WHO guide for rapid reviews, as Andrea mentioned earlier. And we developed our protocol using the Cochrane Handbook systematic reviews of the diagnostic test accuracy studies. And certainly we obtained feedback from the entire team. So as a second step, we developed our literature search with an experienced librarian, and this was also peer-reviewed by another librarian using the PRESS checklist. We searched 3 major databases, and we included studies between December 2019 and September 2021. But due to the time restriction that we had, we couldn't search the gray literature. We did not include any reviews or potential guidelines to search the references. We restricted to English publications with data, and we did not contact any authors for further clarifications or data. Now, as a third step, and similar to what we are doing for a systematic review, we use a predefined form to screen titles and abstracts for level 1 and full text articles for level 2. Before embarking to the screening process, we conducted a training exercise at both levels, and we used our program's online tool, the synthesizer, to perform both levels of screening. However, due to the rapid effort, and nature of this project, we only relied on decisions on a single reviewer at both screening levels. And once this was completed, as a fourth step, we abstracted the data using a standardized data abstraction form. Again, we assessed the form using a training exercise. And due to the rapid nature, again, we made our decisions based on 1 reviewer and 1 verifier. As a fifth step, we aim to perform a risk of bias appraisal of the included studies, but due to the time constraints, we did not proceed to this step. But we certainly plan to perform this assessment once we conduct a full systematic review. We also performed the data analysis and started with the basic descriptive analysis, including country of contact, type of rapid test. Then we assessed the diagnostic accuracy of each index test using a random effects meta-analysis using the bivariate model. And when the eligible studies formed a network of index tests, then we performed diagnostic test accuracy network meta-analysis using the totality of the evidence and accounting for the correlation between tests from the same study. We estimated sensitivity and specificity along with our uncertainty and heterogeneity, and we explored heterogeneity both visually and by investigating potential effect modifiers through subgroup analysis and regression analysis. And for the network meta-analysis, we also assessed the transitivity assumption in its network. So we assessed whether the distribution of effect modifiers was on average similar across the test comparisons. So as a seventh step, we sent our preliminary results to the knowledge users and the entire team and requested for feedback and discussed any implications with the present results. And along with these results, we also provided a one-page summary of our results tailored to the needs of the knowledge user. Then we reported our findings in a detailed document. That was the report. We followed the PRISMA, again, reporting guidelines to ensure transparency and complete reporting of our findings. As you can see in these networks, we included evaluations of 36 different rapid antigen tests. These were informed by 68 studies and approximately 100,000 participants. And the second network includes 23 rapid molecular tests informed by 27 studies and approximately 10,000 participants. So using the whole information, we performed a random effects DTA-NMA. Now, in its network meta-analysis, we compared simultaneously all these index tests. So we borrowed strength from the included studies, and this can also increase precision in the estimates. And our first DTA-NMA suggested that the Express Rapid Molecular Test by Cepheid fulfilled the WHO minimum performance requirements and had the highest sensitivity and specificity. Among the rapid antigen tests, the COVID viral test was associated with the highest sensitivity and specificity, and this also fulfilled the minimum WHO requirements. But results across the studies assessing these tests varied, and this might be attributed to the differences in the clinical and methodological characteristics. So certainly this should be explored further. Now, regarding the challenges and limitations, one of the huge challenges when we worked on the COVID-19 topic during the pandemic was that despite our searches were conducted just 7 months ago, our project is considered outdated already. And another important issue to consider is that our team was hit by COVID-19 while working on the project, and we experienced staff shortages. So in this rapidly moving area, it is possible that we might have missed also studies that contribute to this example, to this review. So for example, the BTNX test that was approved for use in Canada, it was approved in March 2022. But our search was conducted in September 2021. So it is likely that there were no published studies at that time with this commercial project. And we also have other study limitations. I'm not going to go through all of these, but certainly we need to conduct a more comprehensive systematic review and to update it on a continuous basis. So the opportunities, first of all, is that we were able to build our collaborations fast, readjust the team quickly, and we were able to initiate and submit these results to the knowledge users within 8 months and within the requested time frame. We registered with Prospero. We were able to report our results with transparency and accountability, and we also involved 2 patient partners in this work to ensure that patient perspectives are also integrated into our research question. I would like to acknowledge all our collaborations at this point with the study, and we thank everyone for attending this webinar today, and we're happy to answer any questions.
Thank you, Arji and Andrea. That was great talk. So just one question from me to Arji and Andrea. So do you have a sense of how different, if at all, the results would have been if it was done as a full systematic review, and how much longer that might have taken?
Andrea, should I take this question or okay? So I think certainly we would have done much more greater job here if we conduct a systematic review. As we said, we would for sure assess for the risk of bias, that we need to know what is the methodological quality, and in the included studies, it's an important factor. It would have taken approximately, I guess, more than a year or 1.5 years. So in order to be able to do all the steps appropriately, I guess. So I don't know, Andrea, if you want to add anything on that. No. And certainly we would explore more effect modifiers in the analysis than what we have done so far.
Yeah. Just one more question quickly from the chat. So what was an acceptable heterogeneity for the rapid review on COVID diagnostics kit?
That's a very good question. So I'm not sure what is the acceptable cutoff. I don't know if there is a cutoff value that I would say, but certainly when we see heterogeneity, we need to explore that. We need to see what are the reasons for seeing this variation in the effect estimates. So there is no, I guess, cutoff value that I would suggest on the between study variance as itself, because we don't really use an I-square to estimate heterogeneity, to quantify actually heterogeneity in diagnostic test accuracy studies.